Hello, I'm Hiruni Sandunika, representative of today's session, which is about adaptive immunity, and I'm here to representing the Biotechnology Association (SBA). As you can see, this is our immune system. You all know that our immune system composed of two arms as innate immunity and adaptive immunity. So our first naive arm, which is also called innate immunity, do the job of early defense for foreign substances and accelerate our adaptive immunity arm. So this innate arm acts as an agent who represent that there are some foreigns inside our body to adaptive immunity. So innate arm send this message of emergency to adaptive arm while do the fighting against those foreign substances. Right. So like that way, innate cells accelerate our adaptive arm for further activation to fight against foreign substances. Simply, our innate arm composed of immune cells like mast cells, dendritic cells, natural killer cells, complement proteins likewise. So when pathogens enter to our body via epithelial barriers, these cells identify that pathogen and antigen present in cells, engulf them and digest them via different enzymes. Then the MHC receptors represent pathogen peptides to the environment. Likewise, all the cells recognize foreign pathogen and release chemokines like interferon gamma, TNF alpha, and interleukins to activate adaptive immunity. On the other hand, the existing natural killer cells, which are circulating here and there, find pathogens and directly kill them. So, this is the process that happens when pathogens enter to our body. Our adaptive immune system composed of two types of arms like humoral immunity and cell-mediated immunity. This humoral immunity mainly mediated by antibodies while cell-mediated immunity mediated by T cells. If a pathogen entered to our body and then our innate arm recognized it within few minutes to hours, then it accelerates adaptive immunity. Right? When extracellular microbes reach to the humoral immunity, B lymphocytes identify those antigens. As you all know, B lymphocytes can produce antibodies after they become activated because of the innate arm responses. So, after these B lymphocytes receive signals, began to secrete antibodies. So, these released antibodies go and bind with pathogens and block infections and eliminate extracellular microbes. This is what happened in antibody-mediated immunity. When in cell-mediated immunity, macrophages that show microbial peptides on its surface MHC receptors identified by T cells. When antigen present in cells like macrophages engulf microbes in innate arm, they engulf that microbe and digest using their internal enzymes. So after the digestion, microbial peptides can be shown to the external environment via these MHC molecules. Right? After this happens, T cells identify those peptides and activate. Then these activated helper T lymphocytes eliminate the phagocytosed microbes, and in the same time when intracellular microbes like viruses replicate within infected cells, and these cells also can be recognized by cytotoxic T lymphocytes. Thereafter they can kill infected cells and eliminate that infection. That is what happened inside these humoral and cell mediated immunities, right? When it comes to the properties of adaptive immune system, their response have different properties like specificity and diversity, memory, clonal expansion, specialization, contraction and homeostasis, non-reactive to self-antigens likewise. Right? So, if we say what this specificity and diversity means, our adaptive immune system have ability to respond to a large variety of antigens, that is diversity, and also it ensures that distincting antigens elicit specific responses, which is called specificity. Our adaptive immune systems generates responses that are optimal for defense against different types of microbes, that is specialization, and also it allows immunity system to respond to newly encountered antigens, and also our adaptive immunity prevents injury to the host during responses to foreign antigens. right? This image shows an important feature of our adaptive immunity, clonal selection. In here, mature lymphocytes with receptors for many antigens develop before encountering these antigens. In here, a clone refers to a population of lymphocytes with identical antigen receptors and therefore specifies all these cells are presumably derived from one precursor cells. So in here, each X and Y antigen select a pre-existing clone of specific lymphocytes and stimulates the proliferation and differentiation of that clone. This image shows only B lymphocytes giving rise to antibody secreting cells, but the same principle applies to T lymphocytes as well. So this image also shows another feature of adaptive immunity, that is memory. In here there is also X and Y antigens which induce the production of different antibodies via B lymphocytes. Then the secondary response to X antigen is more large and rapid than the primary response, and it is different from the primary response to antigen Y. So antibody levels decline with the time after each immunization process. So the same thing happens with the T lymphocytes when it responds to antigens. So if our body reacts to this first exposure to antigen, which is primary immune response, which is initiated by naive T lymphocytes, and that antigens keep in mind by our immune system, when for the second time same antigen enter to our body, lead to responses called secondary immune responses, where it can eliminate the infection more fastly than primary immune response. The term memory arose as these cells must remember previous encounter with antigen, since they respond better upon subsequent encounters, right? Now we will see what are the phases of adaptive immune responses. So our adaptive immune responses consist of distinct phases. First three are recognition of antigen, activation of lymphocytes, and elimination of antigen, like things, right? First, the recognition of antigen and activation of lymphocytes and elimination of antigen, right? The responses declines as antigen stimulated lymphocyte die by homeostasis, and the antigen specific cells that survive are responsible for memory. Duration of each phase may vary in different immune responses. This is the process, that means antigen recognition in the adaptive immune system. So this is the B cell receptor recognition, and this is the T cell recognition process. So now it is come to the end of this session. So these are the references that I used to extract informations to do this presentation. You can refer these things to polish your knowledge on adaptive immune system. So if you have any queries related to today's topic, you can leave a comment in the comment section. And thank you all for your time. Thank you.